pleasure to introduce Eric Hegg today as our speaker. And Eric is in the Department of Biochemistry and, is it molecular biology?
Molecular biology.
And here at Michigan State. And I had the pleasure of getting to know Eric a little bit because really for a couple of reasons. One is that we were both in something called the Academic Leadership Program, and I was in it a year earlier than Eric, but we had a little bit of overlap there, which I think is when we first met. And I enjoyed conversations with him regarding the Great Lakes Bioenergy Research Center, for which Eric serves as the principal investigator. And he's developed more interests in lignin, which has more overlap with trees and forestry. So I'm excited to hear some things about his recent research and his interests. A little bit of background on his bio. He got a BA degree from Kalamazoo College. So can I assume that are you a native Michigander?
I am not.
You're not. Okay, so you came into the state.
Just for Kalamazoo.
For Kalamazoo. And then moved on to the University of Wisconsin for his PhD. He did a postdoc at the University of Minnesota, so kind of hanging around a lot of the Big Ten,
right.
Great Lakes institutions. And then started a faculty position at University of Utah and gave up the mountains and sun to take a faculty position at Michigan State, and we're really glad that you did and that you're here. So thanks for being here today.
All righty. Well, thank you. It is always a pleasure for me to have the opportunity to talk about my work, and especially this particular topic, because it's something that I really am quite passionate about. Talking about what the work going on in the Great Lakes Bioenergy Research Center, as well as how my own research fits into it. And so when I first when Moshe first asked me to talk here, what she recommended was that I start off with kind of an overview for the first half, an overview of the work going on in the Great Lakes Bioenergy Research Center, or GLBRC. If you hear me say that, that's what I'm talking about. Yeah, I know it's a horrible acronym, but that's what we have. So GLBRC. And just give you an overview of the type of research going on. And then for the last half, sort of talk about how my own personal research fits into that. And so that's what I'm going to do. The first half is just going to be a broad overview. I'll be telling you about what's going on in the center. It's not specific to me. It's not even necessarily specific to Michigan State University necessarily. Okay, so just to start off, what is the Great Lakes Bioenergy Research Center? And it's a research center funded by the Department of Energy, which is administratively housed at the University of Wisconsin in Madison. So they're the primary institution, with Michigan State University as a primary partner. And our goal is to convert lignocellulosic biomass into biofuels. And so we'll talk more about that here in just one moment. But first, I just want to point out that sort of Rich said I'm the PI in the grant, and that's sort of true. I'm the PI on the MSU portion of the grant, but I am not the overall project lead of this. That actually is Tim Donahue at the University of Wisconsin. We also have two scientific directors who are very, very important, Bob Landick and Phil Robertson, one at each campus, who also help oversee the overall science mission of the center. My sort of official title here, if you don't want to use sort of the project lead, is the Director of Business Operations. And what that basically means is I help make sure everything runs smoothly. Because I myself am not doing all the research. There's about 23 faculty members here on campus who are involved in the center. And of course, a number of graduate students and postdocs doing work in those labs. And there's an equal or greater number of faculty at the University of Wisconsin. And then we also have a couple of people scattered throughout the continent, actually. One at the University of British Columbia, a couple at Texas A&M, and somebody at Michigan Tech. So we span a broad region of North America, but mostly we are constrained here within the Great Lakes region, hence the name. All right, and so with that background, what is the vision? What is it that the DOE wants us to do? Well, our goal is to develop novel biological solutions for the production of specialty biofuels and bioproducts from plants. That sounds rather straightforward, but of course this is the Department of Energy we're talking about, and so there's some caveats with this. Okay, they define things somewhat differently than the rest of us might define them. By plants, what they really mean is non-food grade plants. All right? They do not want us competing with food. They don't want a food versus fuel debate, which you may have heard. So it's very important that we stay away from any food plants. All right? Biofuels, they define as, at the moment anyway, anything other than ethanol. All right? So they do not want us going for corn ethanol, for instance. Okay? We already know how to make corn ethanol. We don't need more research into it. And it's got the problem of course, this food versus fuel debate, which we want to stay away from. And by bioproducts, what they mean is anything that you would have formed from a petroleum-based system normally. So plastics, things like that. They're not talking about nutraceuticals. They're not talking about drugs. They're talking about specifically some sort of commodity that you would have made from petroleum previously. All right? So that's what our goal is. We want to stay away from food, and we want to stay away from good agricultural land. And we'll talk more about that later, talking about our work on what we call marginal lands. And we want to take things that we would otherwise be making from petroleum and make them through plant products. Okay, so that's the big idea. And as you might guess, this is a multidisciplinary research center. Right? There's lots of people who are involved in making this happen. And so if we were to sort of look at it on a sort of schematically on a big scale, it might look something like this, where first we want to make sure we develop sustainable cropping systems. Right? Sustainability is very important within the center, and you'll hear me say it multiple times throughout the talk today. So we not only do want to improve the cropping systems, we want to make sure they're sustainable. Then we want to efficiently convert that biomass into bioproducts. And so we'll talk about that. And then we've got some areas which we call the field-to-product integration, because one can imagine working in individual silos in these various areas, but ultimately they all have to fit together. And it turns out it's a lot more complicated fitting everything together than just working on each individual little piece. And we'll talk a little bit about that later. And so to do this, again, we need to build this sort of cross-country, cross-cutting and integrated research teams. And so that's what we've done. We've got some teams that are working primarily on this part of it. We have other teams that are primarily working on this part of it. And then we have other teams that are working on the entire field-to-product transition. Now, you might ask, why do we need this complicated system? I mean, what's so difficult about it? And so for those of you not familiar, I just want to take a step back for just one moment and sort of think about the plant cell wall. So we know we want to take plants and we want to break them down into biofuels and bioproducts. And this could be something like switchgrass, which many of you have heard about, or it could be a poplar tree. So that's one thing that my lab particularly is focused on quite a bit. And so how do you take this tree, this log, and turn it into a fuel? And so the difficulty lies if you think about the smaller structure of the plant. All right? So there's lots and lots of sugars in plant cell walls, but the problem is they're really hard to get out. All right? So if you go down to the very smallest level, you might see a bunch of glucose molecules. So this would be the cellulose. The problem is, and it's very repeating, so that from that standpoint, it's very easy. One enzyme, in theory, can get rid of a lot of, can break off a lot of individual glucose molecules. But the problem is, because of this regularity and the way it kind of can stack on each other, cellulose tends to be highly crystalline. And because it's highly crystalline, it's really hard for the enzymes to be able to get in and break apart the cellulose into the individual monomers. Then you've got the cellulose, which is sort of wrapped around by this molecule known as hemicellulose. And hemicellulose is simply a fancy term for lots of different sugar monomers linked together in lots of different ways. All right? It can include glucose, but it can include xylose, mannose, arabinose, and exactly its structure depends entirely on the plant you're talking about. So that adds complication, yet additional complications, not like sort of one structure of hemicellulose. And this is sort of wrapped around this crystalline material, making it even harder to access the glucose, which is especially what we really want to get to. And then we've got this, you can sort of shown here in sort of these red lines, sort of wrapped around everything is this molecule called lignin. And what is lignin? Well, lignin is something else that provides rigidity, among other things, as well as protection from the plant from breaking down the cell wall. After all, we want to break down the cell wall to release the sugars. The microbes that are out in the world would love to break down plant cell walls to release the sugars. The plants, of course, do not want to be broken down to release their sugars, right? They are trying to fight us. And one of the ways they fight us is by using this molecule called lignin. So what is lignin? Well, lignin is a polymer made up of typically around 3 different monomeric units shown here. And they are put together in different ways with lots of different linkages in what's known as sort of a radical coupling process. And suffice it to say, it's a complicated process where if you can imagine it, it can pretty much happen. Right? So there's no regular structure of lignin. It's sort of like a snowflake. No two things are alike. Right? So it makes it very, very difficult. And if you do manage to break this down, what you have are a bunch of aromatics. And aromatics, as you might have guessed, aren't particularly healthy for most microbes. Right? So it's another way that the plant has devised to make it very difficult for us to break it down. So now we've got this conundrum. We have this great material. It's got lots and lots of energy. It's got lots of sugars. It's got, of course, all this lignin. And, of course, lignin is, on the one hand, bad. On the other hand, there's a lot of carbon-hydrogen bonds. And, in fact, if we break it down, you could almost imagine that to look like a fuel itself or the beginning of a polymer, such as plastics. All right? So we have this perfect material in many ways, but it's really hard to access. So what do? Well, we put together this multi-investigator team to help tackle this problem, because no one person or one lab alone can possibly have the expertise to deal with this intractable problem. And so, as I've already mentioned, we have it broken up into teams. There are 3 teams which are dealing with this first half, starting from the lands, and I pointed out again the marginal lands, not the normal food crop land, and getting lots and lots of biomass from that can be easily broken down. Then there's another group of teams who are involved with taking this and then converting into the bioproducts. And then again, we have these teams which really go from the beginning to the end. And what I'm going to do is just very, very briefly tell you about what each of these teams is up to. All right, so if you think about this intractable problem, how are we going to take this biomass and break it down? There's a couple different ways one can think about approaching it, and one of them is to make it easier to break down the biomass. And so there's 2 approaches that the GLBRC is taking, at least 2, I should say. The first is engineering plants with lignin that can be more easily deconstructed. So it turns out that by a great collaboration by people here at MSU, UW-Wisconsin, and the University of British Columbia, they identified genes which allowed them to put in special monomers into lignin that produce, I don't know if you can see it here, this is an ester linkage. So instead of the typical ether or carbon-carbon linkage that you see in lignin, you've got some number of ester linkages. So if you remember your organic chemistry, esters are relatively easy to hydrolyze under basic conditions. All right? So in essence, they've made this lignin easy to, and we call it unzip, okay, simply by adding base and breaking these ester linkages. All right? So that's one approach. And I'll talk more about that later with my own particular research. Another way is to engineer the plants with improved polysaccharides. In other words, the sugars that are more easily broken down, those that don't form such a nice crystalline structure, known as MLGs, or mixed linked glucans. Right? So again, it's glucose, but it's glucose that's put together in a different way that doesn't allow it to form such a crystalline material. It makes it easier for the enzymes to release the sugars. And so those are some of the things going on in that particular team. Another team is looking specifically at switchgrass, trying to improve the productivity. So not only do we want to make it easier to break the plants down, we also, of course, want to improve the amount of carbon that we can have converted into biomass, so CO2 into biomass, on these marginal lands. And when you talk about marginal lands, you have to think about things which will almost certainly cause stress to the plants. We're talking about nitrogen deprivation, right? They're not particularly fertile soils with lots of ammonia, maybe, or some other nitrogen source. Often they are water stressed, all right? So these are common stresses. If the land didn't have these problems, they wouldn't be marginal. We'd be farming them already. All right? So that's what we mean by marginal lands. Lands which, for whatever reason, are not productive for farming. It can be because it's on a really steep hill, but often it's because the soil properties in some way don't lend themselves to really good, a really productive soil, all right, for farming. And so, so one of the things we want to try and understand is how we can improve the productivity on these marginal lands, especially when they're under stress, such as this nitrogen stress I already mentioned, drought stress, or I haven't mentioned it yet, but cold stress. All right, there's a lot, there are a number of different types of plant species that can grow very well and very fast down south, but how do we move that up this latitudinal gradient up into the Great Lakes region? All right? What are the genes that allow the things that can normally grow up here and be cold tolerant? And can we then introduce them either by breeding or through molecular biology into some other plants that would give us sort of the optimal traits that we're looking for? And so there's a lot of research going on in this area with this team trying to understand how can we make the plants fix carbon dioxide more productively for a longer period of time and yet still not be killed when winter time comes here in Michigan. Okay? We have another important team who's interested in environmental performance. All right? So as I mentioned, sustainability is a very, very important component to our research, and I'll keep coming back to it. And this, what we care about is not only how much biomass we can grow, but whether or not we can do this sustainably for the next 50 years, for the next 100 years. What's happening to the soil? What's happening to the atmosphere? For instance, how do these different cropping systems impact the ability to sequester carbon dioxide, not only in the biomass itself, but in the root system, right? Sort of literally sequester it down back to from whence it came, if you will, down underground. Another important thing, we talk about fertilization, so nitrogen fixation is obviously going to be critical, right? How can we improve the nitrogen fixation in the soil that we're looking at? But also what's important is N2o emission. So those of you not familiar, N2o, or nitrous oxide, is a gas which is naturally produced in agricultural systems. When you fertilize, you've got a little bit of excess fertilizer. What can often happen is first it gets oxidized to nitrate and nitrite. Now that can leach out, and that can end up in the rivers and the streams. You get these giant dead zones, you get eutrophication. You guys have all heard about that, I'm sure. But another process you may not be familiar with is denitrification. And so what happens then is under anaerobic conditions, what often happens is these microbes actually dump electrons into nitrate and nitrite. It's known as anaerobic metabolism, and or anaerobic respiration, excuse me. And along the path of converting it back to N2, N2o is often released. Why do we care about N2o? It's a potent greenhouse gas, and it's also an important gas in ozone depletion. Right? So this is really a bad gas. In fact, the greenhouse potential is almost 300 times worse than that of CO2. All right? So this is really a bad gas, and we're trying to figure out how to mitigate the release of gases such as this while increasing the amount of carbon sequestration underground. Okay? So these are some of the things that the environmental performance team is interested in. Okay, so now we've got the plant system that we want to use, the cropping system. Now we want to break it down. How are we going to do that? And so we've got a number of teams looking at breaking it down and converting it to a biofuel, bioproduct. One of them is looking at sort of what we call the deconstruction and enzyme improvement team. And there's two ways to think about breaking down the biomass. One is to do it sort of chemically, sort of to release these sugars, and then do enzymatic catalysis, which then finishes the release of the sugars by breaking down, breaking the sugar units. And I'll talk a little bit more about that later. Another way to think about it is doing it 100% chemically, if you will. And actually, we got a very neat system which was designed at the University of Wisconsin that the center is really focusing on, known as GVL, or gamma valerolactone. And the idea of this is that basically you can dissolve the biomass, and in doing so, if you choose just the right conditions, you can get a separate a glucose stream, a xylose stream, and then a lignin stream. And you can, depending on how you do it, you can either run this so you don't need enzymes at all, or you just need a few enzymes to break it down further into the sugars. So deconstruction is a very, very important area of our research. But now comes the really interesting part. Because once you have it deconstructed and you've got the sugars, you make this material known as hydrolysate, you want to be able to turn it into a fuel or a product. And how do you do that? Well, we all know about fermentation. We know about making beer, right? We all have, well, maybe not all. Okay, many of us have had the opportunity to make beer, brew beer, and we all have a general idea of how that works. You ferment the sugars into ethanol. We can also ferment into other biofuels and bioproducts to stay away from the ethanol, as I mentioned earlier on. But normally, what the microbes want to do when they see a lot of sugar is they want to grow. Right? They want to create other cells. They want to proliferate. And so they want to take all this sugar and turn it into biomass. And that's not what we want to do. So we can choose the conditions appropriately. So we encourage it to, instead of forming biomass, it takes most of it and converts it into, say, IBA or isobutyl alcohol, or MBO, methyl-butyl alcohol. Okay? So these are some good biofuels, and we have a pretty good idea of how we can change the metabolic network to encourage the microbes to take the sugars and convert it into these sorts of products. But this is where it gets complicated, because we can do this, many people can do this, if you give it pure sugars. The problem is this hydrolysate, this material that you get from plants, is not pure glucose and xylose. It's got other stuff in it. It's got what we call the lignotoxins in it. Remember when I said what lignin was made of? It was made up all those aromatic polymer, and if you break it up, you get these monomers, and most microbes don't like aromatic species. So you've got this and all sorts of other stuff, and what happens is these microbes are under a tremendous amount of stress. And so they don't do what we want them to do. They turn on their stress responses, and they do other things to defend against that stress. And so what we need to do is understand what they're doing. We know why they're doing it. We don't necessarily understand how they're doing it. We want to better understand what these stresses are so we can mitigate them, so we can remove those stresses and allow these microbes to convert it into the species that we want, the biofuels and bioproducts. Okay? So there's a lot of research going on in this. It seems straightforward, and I assure you it is anything but. So now we've gone through the fermentation, and we've made some sort of biofuel, bioproduct. That's great, but we still have a lot of leftover stuff. All right, what's that leftover stuff? Among other things, what that leftover stuff is what we call conversion residue. This can be the leftover cell mass, right, after you have all the cells that did the fermentation for you. It can also be organic acids that may have gotten released during the fermentation process as well. Turns out that can equal a large amount of the overall carbon. And we cannot afford to throw away any extra carbon, not if we want to get the cost of these biofuels down to what you might pay at the pump for normal gasoline. And so if we can't afford to throw it away, what are we going to do with this, what we call conversion residue? Well, one thing we can do is we can burn it, and that has some value to it. You can generate electricity from that, for instance. And so that has some value. But what if you could turn it into, you could find microbes that would basically eat the leftovers, if you will. All right? They would go through the trash can and take over all the leftovers the other microbes didn't want. And so that's what we're doing. We're looking especially at Rhodobacter and Streptomyces as acting as sort of your garbage, your garbage dump, if you will, where they're going through and they're going to find whatever's left over and convert it into something useful. And so there's a team looking at that. Okay, now I'm going to, so I've told you about these teams right here. I'm only going to very quickly go through kind of these cross-cutting teams. But I think it's important to just for you to sort of see the whole general picture now. All right, one of the things that's really important is what we call this multi-scale modeling. All right, what does that mean? It means we want to understand how this whole system is going to be put together. We can model, sort of the farming landscape on the very big scale. Right? Now, I'm not talking just an individual farmer's field. I mean, the large scale. What does that look like? And we can model how a biorefinery might work. But these things don't work in isolation, right? They have to work together. Because the feedstock demand, as determined by the biorefinery, is going to affect the price and availability. Likewise, the price and availability is going to affect the demand. And so these things are all integrated. And somehow you have to get the stuff by stuff, I mean the plant material. You have to get the plant material from the farmer's field or from the, if you're looking at a poplar plantation or whatever, from there to the biorefinery. It has to get there somehow. How are you going to do that? And so all this stuff has to fit together, and we have to understand the trade-offs. Right? This is part of not only the environmental sustainability, but very important, the economic sustainability. The reality is we may wish it were otherwise, but no one is going to right now today go out and spend $5, $6 a gallon for gas if they can fill up for $3 a gallon. There may be 10 people in all of the East Lansing area that will be willing to do that. Most of the people won't do it. And so we have to figure out how we're going to make it economically viable for the farmers, for the landowners, as well as for the people who own the biorefineries. We also have to think about the complications of turning feedstocks in the real world into fuel. We can, in the lab, do all sorts of things. In the greenhouse, we can grow up plants, and they're very, very reproducible, and they look the same all the time. But in the real world, the plants don't always look the same from year to year. It's not just that you might have switchgrass one day and poplar another day. From year to year, from month to month, the individual switchgrass might not look the same. There are lots of things going on. There's droughts, there's floods, there's heat, all these stresses. There's various pathogens, such as pests, which will attack it. And the plants have to be able to respond to all of these things. And they've developed an incredible way to respond to this, but sometimes in that response, they form things known as secondary metabolites. These molecules that help them defend against some sort of pest invader or something else, or deal with some stress. But that can affect the microbes' ability to then break down the sugars that come from this. And in fact, we've seen an example where we can take this is called switchgrass hydrolysate, and this is what we see under normal conditions. We can look at how well the plant, or the microbes rather, can grow. And then we had this drought year. And you can see here there's almost no growth on the hydrolysate in the drought year. All right, so clearly there's something different going on in the plants when they are under a drought stress. And this is the real world that we live in. We can't stop there from being drought. And in fact, if anything, we have to get used to more wild swings between lots of rain and no rain in the new climate cycle than we have had in the past. So this is going to continue to be a real problem. We have to understand what's going on and how to mitigate it. And finally, this one cross-cutting team, which is near and dear to my heart, and I'll talk a little bit about the research going on in my lab later, has to do with lignin valorization. In other words, taking that lignin, remember that awful material I told you about, which was, the microbes didn't like, it was toxic to the microbes. It turns out, though, there's a lot of carbon there. And in fact, upwards, say, of the poplar, upwards of 25% of the poplar carbon can be lignin, and in fact, closer to 40 to 45% of the total energy content of the poplar can be because of lignin. So we simply can't throw it away and say we're not going to use it. What do they do right now in the pulp and paper industry? Generally, they burn it, all right? And that has some value. You can burn it. But we believe that you can do something better with it. All right, we believe that we can take these various lignin streams, we believe we can depolymerize them down into their individual units, and then we can begin to build them up into some sort of useful bioproduct. And this is one of the things that Mojgane and I have started to collaborate on, thinking about how we can take the lignin that we generate and turn it into a useful bioproduct. Okay? All right, so that's a little bit longer than I expected to take, but just an overview of the GLBRC and all the different types of research going on, not just at UW, not just at MSU, but at all of the institutions that comprise the Great Lakes Bioenergy Center. And so I thought before I'd go on and sort of dive down a little bit into research sort of specifically that I'm doing related to that, I would just sort of stop and say, are there any questions now, something you don't understand about either why we're doing what we're doing or the big picture or anything along those lines? None? Okay. All right. So I'm going to go ahead and do a deep dive here then, not that deep, but just give you kind of an overview of some of the things that we're doing specifically to address the mission of the Great Lakes Bioenergy. So some number of years ago, in collaboration with David Hodge, he was a faculty member in chemical engineering who, unfortunately for MSU, just left not too long ago to Montana State University. But luckily, we're still collaborating. But he and I started this collaboration where we were trying to find out better ways to pre-treat the biomass. So if you take this poplar log, and sometimes literally we get logs. The first thing we do is we chip it, we break it down into smaller pieces, but then what do you do with those wood chips? And the first thing that one approach is to do what is known as a pretreatment. And what the pretreatment does is it basically loosens up the biomass. It makes it easier for the enzymes to penetrate and release the sugars. And it can do it in a number of different ways. It can decrease the crystallinity of the cellulose. It can also begin to remove the lignin and the hemicellulose so that it's easier for the enzymes to penetrate to get to the cellulose in the first place. And so we developed this system known as copper HP, which simply means that under basic conditions, we mix in hydrogen peroxide. And if you just, under basic conditions, mix in hydrogen peroxide, you have a process known as AHP, or alkaline hydrogen peroxide. And then if you add a small amount of copper catalyst, and specifically we found that copper bipyridine works the best, you have a process that we term copper AHP, or copper catalyzed alkaline hydrogen peroxide. And if you look at the sugar yields that we were able to obtain, what you saw was that with each, the copper AHP was, significantly better than AHP alone or alkali alone, or certainly better than the untreated poplar. Okay, so we're able to release much more of the sugars following enzymatic hydrolysis. All right, and so this was, you can see, about 5 years ago. We've published on this a couple of, a number of different times. Those are two of the references. But there were, we recognized there was still a problem. Right? The problem was there was still a lot of cost pressure points. We could do it, but it was expensive. Why was it expensive? The yield still wasn't very good. It was about 60%, not bad, but not nearly as good as we need to get it. The catalyst copper bipyridine is expensive, not the copper, but the bipyridine part of it is quite expensive. Hydrogen peroxide is expensive, and we needed to use a lot of that. And the enzymes, of course, are expensive. All right? And so the question was, could we increase the yield while simultaneously reducing the inputs? And so the answer is yes, we could. What we did was we put in, we made two modifications. The first is we put in this alkali, what we call a pre-extraction, or a stage 1, where we just first extract the wood in basic conditions. Okay, we remove some of the extractives and some of the lignin, some of the things that would inhibit the further downstream processes. The next change we did was to go to fed batch hydrogen peroxide. And the only exciting thing about this is it means we didn't add it in all at once. We added it in slowly over time so that it could be used more efficiently. Okay, so it wasn't really a particularly insightful change, but it was an important one. And when we did that, we obtained results that look something like this. So here I'm showing you both the glucose and the xylose yields. Here's the reference case, what we were at before. And this is what we did when we added the two changes. And you can see this is also dropping the catalyst concentration from 2 to 1 millimolar. So we've cut the catalyst concentration in half, and yet we've still improved the glucose yields by about 35%. All right? And so that's obviously a really big improvement, and we were very happy about that. And one of the things we were able to show then is we could at the same time then reduce the hydrogen peroxide, that we didn't need to use the full 100 that we were using. We could drop it down to 75 and still get almost the equal amount of glucose yields. The other interesting thing about this, which we found out, was that if you look to see how much lignin you need to remove, once you get about 40% lignin removal, that's about enough. You don't need to remove all the lignin. You just need to remove enough of the lignin so the enzymes can penetrate in. So you can certainly remove more, but you don't have to. So 40% seems to be kind of that magic number of what we want to remove. And so if we go back to this same slide and say, okay, where were we as of a couple of years ago? I would, add this this is where we were before, and we added now that new bar with this new system, and we see this 35% increase, and with half the catalyst concentration. So we're pretty excited about that, but we realized that that wasn't good enough still. All right? We still had some cost pressure points we wanted to address. So a couple strategies we decided to take. One is we said, well, what if we increase the severity? Okay? What if we increase the severity of this relatively inexpensive first step, the alkaline delignification, so that we can then decrease the severity of the second stage, which we're calling the alkaline oxidative post-treatment. In other words, the copper AHP part of it. Okay? So that was the idea. And also the idea was that if we pick these, we sort of find the perfect spot between the two, we might actually generate lignin, which has the optimal properties to be useful for downstream processes. And I will get to that, I promise. So what we did was we increased the temperature from that first step. We were just basically doing this at room temperature, 30 degrees. Barely above room temperature, and then so we heated up to 120 degrees. Still, by an industrial standpoint, still very mild conditions. Okay, 120 degrees, well under what you'd see in the pulp and paper industry, for instance, for something like this. And what we found is that we remove about 15% of that lignin by increasing the temperature. So that's useful. And then if we look to see how that affected the performance, I'll show you some data here. So it's going to take a minute to kind of walk through. These circles right here, these gray circles, this is what it was before, okay, when we were still doing the pre-extraction at room temperature. And you say, well, that looks about the same. And it does. It's only a little bit worse. But look, we've had to add 2 times the amount of enzymes to get to the same glucose yields. All right? So if we take that into account, then the higher temperature definitely decreased the cost. And you can see that it's more clearly here in terms of lowering the enzyme concentration. This is what we had before, and if we lower the concentration of the enzymes very quickly, the amount of sugar for the base case sort of drops down. But for the other case, it actually, you see, we can drop it down about 20, well, actually, what was that, about 30% or so. We can drop down the enzymes without having any negative impact on the amount of the sugar being released. And because enzymes are one of the critical components of the cost, this is a really important improvement. And then finally, again, we've also shown, and again, this is a huge cost point, is the amount of the bipyridine. Can we add less bipyridine? So we're just showing here what happens if you reduce the bipyridine. This is what it is if we do the pre-extraction at room temperature, and this is what happens if we do it at the elevated temperature. And you can see here quite clearly that without having any drop in yield, we can lower the bipyridine concentrations by about a quarter. And so that, again, is a huge cost savings process. So that's the first strategy we're taking. The second strategy is to use these zip lignins. Remember I talked to you about those zip lignins before with these ester linkages? And they break apart very easily under basic conditions. Well, what are we doing with our poplar? We're putting it under basic conditions. So the idea is if we combine the zip lignin poplar with our process, which should be much easier to break down. All right? And so that's the idea. And in fact, what I'm showing you here is some data obtained. This is what happens if you do alkaline-only extraction, okay, nothing else. And this is what happens if you do the copper HP. And you can see, not surprisingly, you get higher glucose yields if you do copper HP. This is wild type. So this is what the normal poplar look like. It's a slightly different poplar line, so you can't compare the numbers exactly, but this is the wild type poplar. And shown in blue is what we call line 7. Line 7 has the zips put into it. All right, so in theory, it should be easier to remove the lignin. And what you can see, in fact, is that we get about a 10% increase between wild type and the line 7 where we have the zips. All right, so clearly, it is easier to remove the lignin. So if it's easier to remove the lignin, is that what's helping to get to the sugar? And so what I'm showing you here is some microscopy that was done in collaboration with Shiyu Ding here on campus. And what I'm going to show you here first on the left in red is the lignin. And what you see here is untreated alkaline copper HP versus wild type versus line 7. You can see in line 7 you can hardly see the lignin anymore. You've just reduced it an enormous amount relative to what we saw at the wild type. So clearly we're removing the lignin. Does that help sugar accessibility? Because our hypothesis is that it does. And in fact, shown here, again, this is wild type on the left, line 7 on the right. What we're doing is we're looking at some fluorescence microscopy here. And what you can see very clearly is that the line 7 is much, much brighter. So in other words, it's being able to access the sugar much more readily than it could in the wild type case. All right? So our hypothesis that if we make it easier to remove the lignin, that we would in fact remove more lignin, and that would then make it easier to access the sugars, in fact, is correct. And so does that allow us to then make it use less stringent conditions to break apart the biomass? And so, in other words, can we lower the chemical inputs? And so what I'm showing you here in green is the wild type, and in red is the zip lignin. And you can see that the zip lignin in all cases is much, much better, gives you higher yields, in other words, of sugar release, regardless of the catalyst concentration or the hydrogen peroxide. And you can see in both cases we can lower them without having a substantial decrease in the yield simply by going to the zip lignins. All right, so making it easier to break down the lignin is in fact a really key component. We can talk about what that might look like in the field later if you're interested. Okay, that pretty much says the same thing, so I'm going to go by that. So I have time to get to this last piece, because I think this last piece is really important. And it's one of the things that got Mojgan and I talking quite a bit over the last year or so. And so we realized as we were doing this that, of course, we were generating a really good way to remove lignin from biomass. And initially, I was thinking just like the pulp and paper industry. So you get the lignin and you burn it. But then we realized, well, that's really stupid. There's got to be something better you can do with the lignin. And you can see how much of the lignin we're removing sometimes. So here's what untreated biomass might look like. It sort of settles in the bottom of this liquid here. If we do the reference case, this is what our normal copper HP. And then if we do those modifications, and so why is it getting smaller? The same amount of biomass was added. It's getting smaller because the lignin and the hemicellulose is getting dissolved and removed. And so we have a lot of lignin now in solution. And so the idea is, well, can we get that lignin back and then do something useful with it? And in fact, we can get it back. All we have to do is drop the pH. If we drop the pH, we get this really beautiful kind of tan color material drops out. It's very different. If any of you have visited a pulp and paper mill and looked at craft lignin, it's this really black oil, tar, nasty sort of stuff. All right, this is very different. All right, this is almost what we'll call native-like lignin. You'll see what I mean here in just a second. But it's this beautiful lignin that we can very, very easily isolate from that liquid that I showed you just a moment ago. All right, so I say that it's native-like. What does that mean, and why is that important? Well, among other things, you can see you can look at the color, and you can see it's not this black tar the way the other stuff is, so that's good. It also means that we haven't put in a lot of other linkages. And so if we analyze this by NMR, this is the untreated poplar lignin, and this is what happens, this is what the lignin looks like by NMR after we remove it using our process and then precipitate it. And you can see that there are some differences, but these differences are basically here and here, okay, and then this as well. And all of these what we call the vanillin. And basically what we're looking at here is we're looking at things that are slightly oxidized, all right? So in other words, if this is what the syringeal unit might look like, and this is what the guaiacol unit might look like, basically what we've done is we've oxidized what used to be an alcohol to a ketone here, all right? That's the biggest change that's gone on. And it's not complete, it's just in certain places we've oxidized it. We don't see evidence for any other linkages, and I don't have time to show you the data, but we've looked for other linkages that got put in during our process. We don't see any. Why is that important? It's important because it means we're not complicated. I mean, let's face it. Oops. Lignin is already complicated enough. We don't want to put in any additional linkages we have to figure out how to break down, right? So it's really important that we haven't done it here. All right, so why else were we excited about this? Not just because of the linkages that we have not put in, but we're also excited because of the slight amount of oxidation, ironically. You say, why might we be interested in the oxidation, taking this alcohol and converting it into ketone? The reason we were excited about it is because there is a class of enzymes that out there in nature known as the Lig enzymes. And there's, you know, it's alphabet soup. I'm not going to go through what each of them does. But basically what this family of enzymes allow the microbes that contain them to do is to take lignin, to first oxidize it from an alcohol to a ketone. Does that sound familiar here? And then take that ketone and via a glutathione-dependent process. A glutathione is just a fancy type of amino acid. It's got a sulfur on it. It attacks, let's see, this carbon right here, and it forms this cross-link between the glutathione, releasing 1/2 of this unit, and then by taking another glutathione, it releases the first as a disulfide to release the other unit. So this is what's already happening naturally in nature. Not very fast, but it does happen. And then what happens is nature takes this disulfide, this glutathione disulfide, and re-reduces it back, and so it can start the whole process over again. Right? So that's what nature does. And so we said, well, you know what? We've already done this first step, going from the alcohol to the ketone. Can we use a small mimic, a small organic thiol that can do the same sort of chemistry? Can we mimic what's going on in nature? And so that was what we wanted to do. And this was actually work done by Gracie Luklinger, sitting right up here in the front. So you can, if you have any questions, ask her how it works. Okay? But basically, the answer is yes, which really does work. So the first thing she did was she synthesized a model dimer. This is not exactly what lignin looks like, but it's a model. It's a simple model where we've already oxidized the alcohol to the ketone. And what she was able to show is very clearly that this material goes away to essentially nothing, and the products that you would expect to see build up over time simply by putting in a small amount of beta-mercaptoethanol. Okay? And it works very, very nicely and cleanly. Then she did another really important experiment, because what we assumed is that this was biomimetic. This was working the same way the enzyme was working. But if that's true, then we should form this thioether intermediate that then has to get broken down further. And in fact, what you can see is when she synthesized what we assumed was the intermediate and then reacted it with beta-mercaptoethanol, that intermediate immediately decayed to zero, and very rapidly you see the production of the expected product. All right? So this is showing not only that it works with these simple systems, but that it works in the way that we predicted it would work. All right. So that's all well and good, but let's face it, that's a relatively simple model that doesn't look a lot like lignin. What about something a little more complicated? So she either synthesized or purchased a lot of different molecules here. I won't go through them all in detail, but suffice it to say she looked at lots of different model complexes, as well as a number of different thiols shown right here that she tried. And one of the things, and I'm just going to sort of give you the bottom line here. The bottom line is, even when you get into these complicated ones, this is starting to look like real lignin. This is, let's see, so this is the SS and the GG, right? And so this is the real linkage, well, albeit oxidized, and it still works, right? So even with what we'll call real lignin dimers, it still works. All right. Now, if you ask, what about all the different thiols one can imagine? If we use glutathione, that's the most obvious question. That's what the enzyme uses. Can we use it? The answer is, not very well. And we think it's because it's too big and bulky to access in this non-enzymatic system to get in where it needs to go. But a lot of these others work very, very well, especially the smaller, the better. All right. And the other thing we learned is that, in fact, this oxidation, just like occurs in the enzyme, is very, very critical. Okay? So those were the big take-home messages. And then that leaves then the final sort of question, well, that's all well and good, but does it work on real lignin? Come on, this is just stuff you made up in the lab. What about real lignin? And so we took some real lignin that we isolated from the copper HP, and if we analyze it by GPC, so here we're just looking at the size of the lignin fragments, and that's shown here in gray. And then if we incubate it either with the thiol DTT or dithiol-3-O-tor or the beta-mercaptoethanol, we get the the chromatograms here shown in either blue or orange. And what you can see is not only is the peak getting literally smaller in size, showing we've got more and more small units and less big units, but even the big unit is getting smaller in size. Right? So we are able to break down real honest-to-goodness lignin. And we're still working, trying to understand exactly what the mechanism is. We know, we suspect it's at least among other things, just like we see with the models, but we think there may be other mechanisms as well. And so we're looking into that. Okay. And so just to summarize here, what I hope I've been able to show you, at least in the research going on in my lab, is that we are able to quite effectively pre-treat real poplar logs, so we can break it down and to release those sugars that can then be fermented into biofuels and bioproducts. And by carefully increasing the severity of the alkaline delignification stage, we're able to improve the process because we can do less severity, which is where the cost is, on the second stage. Okay? We've also shown that we can use this zip lignin poplar and that it works exactly like the people who initially designed it. I wish I could say that was me. That was not me. It was in the center, but it wasn't me personally. But it was a great great discovery that they made, and it works just like we think it would work. And that is, it helps us remove the lignin and expose the sugars. And then finally, we're able to show that this lignin stream that we get out, this really nice, clean lignin stream, and it really is clean because we can analyze it for all sorts of sugars and all sorts of other components, and it is very clean, and that it is susceptible to being broken down via an enzymatic-like reaction mechanism to release the individual monomers that can then be built up into some sort of biofuel or bioproduct. And with that, that sort of comes to close. I need to thank the people who helped me do the research that I talked about today, because of course I didn't personally do any of it. It was all done by basically these 4 people on top, 2 postdocs who have since left the lab, really helped get the thing going, along with the graduate student in David Hodges' lab, was there at the very, very beginning. And we owe a lot of debt to him. He's now at Oregon. He's at a biofuel company, DuPont, out in California. Gracie Liu is a graduate student still here with me working on the lignin, so she's here in front. And then my collaborators, I've already mentioned David Hodge. Ned Jackson, I work very closely with this lignin depolymerization. In fact, Gracie is a joint graduate student between me and Ned Jackson in the chemistry department. Shiyu Ding did all the imaging. John Ralph of the University of Wisconsin not only came up with the idea of developing the ZIP lignin, so we owe him a great bit of debt for that, but he also is a real expert in NMR analysis. And so we've worked with him closely. And I unfortunately didn't talk about Siva's work at all. But anyway, and that, of course, we're funded through the Department of Energy. And that's all I have. So thank you. So, did I leave enough time for a couple of questions? Okay. Yeah.
So the poplar that's growing that has the lignin.
Right.
It's easier to break down the lignin. Does that mean that it's easier for biological organisms to break it down?
That was the perfect question, and we wondered exactly the same thing. So we first started growing it in greenhouses, and the answer was no. But of course, we're inside a greenhouse, right? We're not out in the real field. So we have a real field trial going on right now. And the answer appears to be no, at least not in the amount of time that we want the poplar to live. So poplar rotation crops typically are 5 to 7 years for this sort of material. Now, if you wanted this to grow for 20 years, the answer might be yes. At least for up to 2 years, which is where we are right now, the answer is it doesn't appear to have any effect at all. We're hoping to continue the trial for a 3rd and even 4th year potentially. It's not clear we're going to be allowed to do that. The USDA, of course, we have to be very careful with field trials of transgenic trees. And this in particular is a problem because the particular poplar used here is an aspen, which, and I didn't know this, but it propagates through root suckers, so it makes it extra hard to contain. So they're really concerned about containment. So whether or not they're going to allow us to keep this trial going forward, I don't know. But at least for 2 years, the answer is surprisingly no. It doesn't make it easier by biological. But it's very surprising. Yeah, great question. Anything else? All righty. Oh, yeah.
You hit on the food versus fuel.
Yes.
And I guess I was wondering, so marginal lands have often been a refugia for early successional conservation and kind of been the only preventative for agricultural advancement into those lands.
Right.
Now we're saying that we're going to devote that to biofuel production. And I just wonder what the center would comment on that.
Yeah, so that's really a great point, right? Because there's only sort of so much land, and what are you going to do with it? And a lot of people, this is outside my area of expertise, so I'm going to give you the center answer as I understand it, recognizing it's not my particular area of expertise. But you are correct. I've asked similar sorts of questions. A lot of the land, for instance, is this RCP, Reserve Conservation, CRP, thank you, land that you could imagine coming out. I mean, a lot of these 10-year or 20-year, however long these leases are coming up. And you could imagine it coming out. And that may be problematic. So what is the center thinking about? One of the things we're thinking about is trying to stay away from annual crops. So perennials, that's why the switchgrass, the poplar, mixed, I didn't talk about it all, but mixed prairie. So that we get, especially with the mixed prairie, you get these ecosystem services while at the same time basically being able to mow your field and not have any of the adverse effects that you might have with what might otherwise be intensive agricultural processing. The other thing that this is also why they want to get away from fertilizing, right? Not only for the cost, but again, this environmental impact that it would have. So these are the things that they're looking at, and that's why the people who are really into sustainability are really focusing heavily on the mixed prairie. But the other thing they found, and this is, I think, it certainly surprised me, I can't say it surprised everybody, it surprised me, is that you can have monoculture as long as on a small scale. So you can still have good ecosystem services. In other words, what you don't want is something to look like Iowa or Illinois, where you just have nothing but corn, right? But you can have a big field of switchgrass and another big field of something else, as long as they're sort of, they're mixed up, right? You don't have this overall, and that the ecosystem services you get from that, I'm not going to say they're as good as if you had sort of a mixed prairie, but that they are much, much better anyway than you might expect from what would otherwise be sort of monoculture farming. So these are all the things that they're looking at and thinking about. Nobody has the right answer, the silver bullet. Almost certainly the silver bullet, well, there is no silver bullet. The best answer is going to be dependent on the region that you're in. How marginal is the land we're talking about? Why is it marginal? Can you get away without doing any fertilization? What does the mixed prairie look like in that sort of area? Can we make it so that so that the switchgrass can have really late flowering time, so you get a lot of biomass, but still survives the Michigan and Wisconsin winters? These are all things that have to get worked out. But you bring up a really important point, and I'd be lying if I said we had all the answers. This is one of the active areas of research, because there's a lot of people out at Kellogg Biological Station that this is what they do, and this is what they think about day in and day out. And so they're very passionate about it. Yeah.
Like residue, like corn stover?
Right. So residue can be used, and in fact, it has been used. And it turns out that right now, residue is already has a purpose. It is being fed to cattle, among other things. And you do want to till in a certain amount. How much you need to till back into the soil depends on the soil. The amount in Illinois, in Iowa, is less than the amount here in Michigan, where we don't have as fertile a soil. So, but you can take the residue. And for the first number of years of the center, we did a lot of stuff with corn stover. And a lot of people are looking at wheat straw and things like that. So I think that the Department of Energy wants us to move away from that a little bit, because they think part of those things have already been solved, and they want us to think about sort of these other more dedicated crops. But definitely people are thinking about these residues. But we have to remember, it's not that they have no value now. They are being used. Maybe not as efficiently as they could be, but they are being used. But that is absolutely a possibility. Yes. Yeah.
So it seems like you've made good progress in terms of the glucose yield
Mm-hmm.
and in the past few years. So how close would this be to commercialization? And I guess the related question is it
Right.
economically
Viable? Yeah.
And could it compete with fossil fuels?
Right. So the answer at the moment is no, it can't. But I don't think we're necessarily, depending on what happens, we're not necessarily as in the political scheme. We may not be as far away as you might think. So if you were to think about the whole, it also depends on how you do the numbers, right? Do you just say, what can I make the fuel for? And you ignore the opportunity cost, right? So we just did sort of a techno-economic analysis with Chris Saffron over in the engineering department. We're also working with Brian Balls out at MBI to ask these sorts of questions. How far along are we? And we're still probably $3 or $4 a gallon above what we need to be. I think the most recent numbers came in at actually surprisingly low until I found out the assumptions, and that explained why it was so low. I think it came in at like $5 a gallon, which I'm like, oh, hey, that's pretty good. We're not that far. You know, if oil gets back up to $120 a gallon, I mean, it wasn't that long ago, if you guys remember, we were spending over $4 a gallon, $4.50. We had to get a loan to fill up our car with gas. I mean, it was incredible, right? And then, of course, the economy tanked. Then they found, you know, fracking, and they got this natural gas, and that also put, you know, downward pressure on everything. So what are we sitting now? About $50 a barrel, or something like that, 50, 60? Anyway, it's less than half of what it was in the peak. So if we go back to the peak, which is outside the realm of possibility at some point, you know, gas jumps back up again, you know, we're looking better. But to get to that $5-ish, some of the assumptions, they ignored opportunity costs. So the opportunity cost, to those of you who aren't sort of economists, I've learned is you know, no one's gonna put down money for $100 million biorefinery if they're not certain they're gonna get some return on that, right? They might as well just put it in the bank or something that's safer. So they have to get more back than they put in, or they're not gonna invest in it. So that's the opportunity cost. So you have to sell below, you know, what it would otherwise be. And they made some other assumptions. But my guess is we're probably about double what we need to be right now. So we're not outside the realm of possibility, but we still clearly have, you know, another decade to go, I would say, unless something happens with the gas prices, right? But if gas prices stay where they are right now, it's probably a decade to go, I would say. And also, it'll depend a lot on what the other fuels, right? I mean, we did this into ethanol, but we've got this problem with ethanol, this sort of the ethanol blend wall that you may have heard about, right? Where people, unless you have a certain flex car, you know, they don't want to put more than a certain amount of ethanol in because of the extra corrosion in the engine. And right now, we pretty much are producing about as much ethanol as the country wants to consume. Again, it goes back to a choice of the consumer to some extent. But if we find a better, you know, if isobutanol or one of these other ones really takes off, then that's a game changer, because then you could go practically 100%, you know, not 100, but you could go much, much higher percent than right now the 10, 15% they want to sort of limit the ethanol at. So it's a complicated question, lots of moving parts.
All right, I'd like to remind everyone that we have a reception across the hall in 216, and everyone's welcome to come and continue the questions there. So thanks again, Eric.
All right, my pleasure.